Okay, so continuing on with some of the information as far as what's going on with the mechanobiology, with between the cellular component of tissue and the actual tissue components. So we look here at integrin activation and focal adhesion formation. So remember, integrin is that receptor. It's responsible for transmitting mechanical signals from the extracellular matrix to the cell. So again, go back and look at the components of the extracellular matrix. We had talked about, collagen, elastin, the ground substance. That's all the stuff outside of the cell. And again, those two things communicate with each other for basically the health of the tissue and being able to respond to tissue stress, being able to grow, repair, those sorts of things. So an important thing to note that the communication between the integrin receptor and the cell, and the basically taking the information from the extracellular matrix to the cell, happens bidirectionally. Now, typically this happens in an outside in fashion, meaning that basically when tissue gets stressed, and the reason why this is how it mostly happens is because this is what happens in response to tissue stress. Most of the information flows from outside of the cell going in. So this was what basically transmits, again, signals to the cell to get it to respond. However, it could also happen from the inside out as well, and this is what we see with extracellular matrix binding and what we have in regards to mechanotransduction, which is basically what happens, again, if we go back to the beginning of the discussion. So this mechanical force transduction, working with the endocrine system and the other growth factors and gene expression, is what you have with inside out formation. And we'll show a little bit how this happens with the figures from the text, so you could kind of see how this occurs. Now, what basically one of the responsibilities as far as what happens and what allows the connection between the integrin receptor and the actual cell is what's called the formation of a focal adhesion complex. Okay, so the focal adhesion complex forms and anchors integrin to the cytoskeleton. So now remember, the purpose of the cytoskeleton is a couple different things. It maintains cell shape and support. It allows for the cells to move, and it also provides tracks for intracellular signaling pathways. So again, the ability for the cells to signal and create hormone responses when again, in the case of tissue gets exposed to stress. So when we're looking at this focal adhesion complex formation, and this is an important part of this, again, because of the signaling that has to occur. So the focal adhesion complex is basically a group of proteins and adapter molecules that basically combines when integrin gets activated. And what this does is it leads to this clustering and then the connection between the integrin receptor, the extracellular matrix, and the actual cellular component of the tissue. Now, this is going to be strengthened by what's called syndicans. Syndicans are proteoglycans that are involved in cell adhesion. So these are substances that actually create adhesion between cells. They're located on the epithelial cell surface, and they act as co-receptors for fibronectin, growth factors, and cytokines. So these substances allow the formation of this focal adhesion complex. And again, what this does is it responds to cell membrane stress and activates ion channels. So again, the idea here is, again, it provides this connection because when, again, our tissues are exposed to stress, even, again, take exercise again, for instance. I had mentioned this earlier with strength training. So when you strength train, you have certain adaptations that happen in the muscle. The only way those adaptations happen is if that signaling occurs. And this is obviously very important in regards to tissue repair, because we're trying to not only form, good healthy scar tissue, but we're also trying to re-strengthen that tissue. So the next two illustrations are going to kind of show this. And again, I'm concerned more so with the things that I point out. There's going to be some other growth factors and things of that nature. just stay focused on the stuff that I'm pointing out, some of the different structures, so that you just have an overall working idea of kind of some of the more important elements. I don't want you getting yourself too bogged down, because it can be a little overwhelming, again, looking at some of these illustrations, because they also talk about a lot of other things that I don't think are immediately important from your standpoint. But again, you should have a pretty good understanding of the actual process. Okay, so I'm going to kind of go through this and go step by step. So this figure that you're seeing here is an example of inside out activation. Okay, so again, remember the objective here is to get the extracellular matrix to bind with the integrin receptor. Okay, and this is again as a part of the mechanotransduction process. Okay, so in this situation, the integrin receptor is not yet bound to the extracellular matrix. Okay, so again, just to kind of orientate yourself to what we're looking at here, here's your integrin receptor. Okay, so you can see that there. Okay, your different subunits, which I'm not so much concerned that you understand the difference between those two, but just know here is this little complex here is your integrin receptor. You look over here, so this whole area out here is the extracellular matrix. You can see that this is a component here, a component of the extracellular matrix, this long strand here. So you see initially it's not bound to the extracellular matrix yet. Okay, it has no connection, though you see A, B, and C. It's showing you how it's gradually building into that process. So you see in B, we're already starting to make the connection to the components in the extracellular matrix. So when we look at this picture, again, this is representative inside out, and this begins as a result of the cytokine receptor basically beginning the process. Okay, and this is coming from the inside part of the cell. So part A you kind of see, and you can see the cytoskeleton formation that gradually happens as well when you're kind of looking at this. So again, I'll pull up the pointer. Okay, so here's your actin cytoskeleton that forms here. So when the signaling occurs, and again, I'm not so much concerned. Don't get yourself so caught up in a lot of these other factors that you see in here, but I would reference you to look at the book and some of the things we've talked about in the past. But essentially, when you get the signaling that's going to occur, you start to get the intracellular recruitment of different proteins that eventually will form that focal adhesion complex. Okay, so that's what you're starting to see as you kind of progress with this process. And what eventually forms, this is your focal adhesion complex here. Okay, so here's your actin cytoskeleton that forms, and essentially what happens is, with this chemical signaling coming from the inside part of the cell, you get the formation of the actin cytoskeleton, and then essentially this focal adhesion complex. You don't have to know these different proteins. Just know that, again, it comes from the intracellular signaling coming from the inside part of the cell going out. Okay, is when you get this formation of this focal adhesion complex via that mechanism. So here's the representation of the outside-in signaling. And again, this is essentially what happens when, again, tissue is placed under stress. So again, tissue is performing some type of physical activity, and what this does is it allows the tissues to kind of adapt and respond structurally through different types of chemical receptors, gene expression, things of that nature. So you notice the difference here when we look at this picture. So we don't have the process of, per se, the formation of the cytoskeleton and that connection with the integrin receptor. You already have that in place. So when you look here, so here you have again, orientate yourself a little bit. Don't get caught up in too many of the different factors and proteins. So here's your integrin receptors here. You see them all kind of lined up. It's already connected to the extracellular matrix. So all of these components, if you kind of look at some of the pictures that we showed earlier from the textbook, Those are your components. That's your extracellular matrix. Down in here, you have the actual cell components of the cell. Here's your actin cytoskeleton here, and here's your focal adhesion complex. So your focal adhesion complex is this whole structure coming across here. So with outside-in activation, when the tissue is put under stress, there's signal transmission that's going to occur, and what's essentially going to happen is this is going to be what allows for the formation of the remodeling enzymes to occur. So that gets to be important when we look at tissue, at the process of tissue healing. When we look at going from repair to remodeling, the formation of those remodeling enzymes as you're actually starting to put the tissue under stress and do different types of activities with thinking about rehabilitation. It also allows for gene expression of the extracellular matrix to be able to help form the components of that as well. So we're just basically working that signal in reverse here, taking information from the outside of the cell working in, and to just help to support that structure and that communication between the extracellular matrix and the actual inside part of the cell. Okay, so here we're going to start to look at basically how this works in regards to tissue, because again, this is our primary concern, looking from the standpoint of injury rehabilitation, injury prevention, kind of what actually is happening with the tissue, so we kind of understand how to stress it and how to basically make it heal. So if we look at the mechanobiology in muscle, so we know that muscle's responsibility is force generation through the process of muscle contraction and the connective tissue in muscle. So muscle has both a contractile and a non-contractile component. Both are very important, because even the non-contractile component facilitates load transmission. So even though it's not actually physically active in the process of creating muscle contraction, it's important to again continue to allow the muscle to withstand stress as stress is being applied to it. So looking here as far as how this works in muscle, so a lot of the processes that we previously talked about. So mechanical stimuli and damage cause the release of specific substances, particularly in the case of the muscle. Something very important is called insulin-like growth factor. So insulin-like growth factor 1 is released, and what that does is helps to regulate protein synthesis and anabolism. So if we look at, again, if we just look at something very basic that I think most of you could probably get right now, is just think about what happens with the concept of strength training and muscle hypertrophy. So when you go to strength train, particularly when a goal is muscle, hypertrophy in a muscle, you go and you're basically causing stress to the muscle. You're actually inducing damage. Now, it's not at a big level where you're injuring the muscle, but that mechanical stimuli of lifting weights produces that micro trauma to the muscle. And the idea is, and we'll talk about how that stress impacts this, but you're releasing different things like this, and you're basically essentially trying to regulate protein synthesis. Most of you have heard that protein is very important in the process of strength training and muscle repair. So these different factors, and particularly insulin-like growth factor, helps to regulate that anabolic process. So there's synergy between the insulin-like growth factor system and integrin. So the integrin receptor and that response from that structure, as we're putting the tissue through this different stress, is what helps to produce this response. So this is very important in muscle remodeling, muscle building. So whether you're looking at this from the standpoint of performance or you're looking at it from the standpoint of repair from injury, it's going to work in both circumstances. So something to consider is when you go back and look at, again, how typically that communication occurs from the outside in. So in regards to tissue stress, that's how this usually happens in muscular tissue. Now, most of your integrin receptors are present at the myotendinous junction, so it's where the muscle and tendon kind of come together. So that's where their prominent location is. We're going to talk about some of the other structures, but primarily the formation of focal adhesion complexes primarily occurs at this location. So what essentially will happen is you get stretch activation of what are called satellite cells, okay? So satellite cells are cells that are basically there to help repair the muscle. They eventually will differentiate into mature skeletal muscle cells, and they basically reside inside the basal lamina of the skeletal muscle. So, and that's the area between the muscle cell membrane and the endomysium, which we'll talk about the different layers of the muscle on the next slide. But basically, any type of muscle stretch, exercise, damage will contribute to activation of the satellite cells, which will then divide and diffuse and basically be responsible for forming the protein synthesis, hypertrophy, and repair that's going to have to occur. So the muscles are very highly influenced by this. It's very important that, number one, we do stress muscles to a degree because we want muscles, and we want to obviously be able to maintain a degree of muscle mass and strength. At the same time, when an injury occurs, so and most of the time, if we're talking about a muscle strain, for instance, or some type of overstretch injury, many times it happens at the musculotendinous junction and usually involves disruption of that focal adhesion complex and the integrin attachments to the extracellular matrix. So that's usually where that injury will occur. And looking at another thing is when you have immobilization. So we know that when muscles are immobilized, atrophy occurs. So, that immobilization-induced atrophy is usually associated with a decrease in the formation of the synthesis of the focal adhesion complex. So again, it's very important that muscles are obviously worked and activated even from that perspective, because again, atrophy can occur, and that will actually produce not only just a change in the size of the muscle, but just in the overall biology of the muscle when you're looking at it. Okay, so here we have some of the structures that we were just talking about on the previous slide. So, again, think of tying into some of the physiology that I had discussed. So if we look here here we have a more representative, like pure anatomical picture. Here's a drawing of it so you can kind of get to see it from both perspectives. So here in this picture, you have your epimysium. Here you have the endomysium, perimysium. It's a little easier to see and understand from this picture because you get to see kind of how the muscle is actually broken down into the individual parts. So here obviously you have the tendon, which we're going to be talking about next. Your epimysium, which is basically the covering of the entire muscle. You have the perimysium, which is deep to that, which surrounds groupings of muscle fibers. So this grouping of muscle fibers is called the fascicle. Within the fascicle, you have your individual fibers, and they are covered by the endomysium. So those are your different layers from the perspective of remodeling. And again, we had mentioned that the satellite cells reside in the endomysium, and those particular cells are responsible for some of the regeneration that would occur in muscle tissue. Biology in the tendon. So obviously here we're talking about where the structure responsible for the muscle inserting into the bone. We had mentioned in class the importance of eccentric exercise with this. So the eccentric exercise helps to reverse some of the negative effects of inappropriate loading on the tendon and what basically happens with that. So again, tendons are responsible for transmission of forces to the skeleton coming from the muscle. They also help with joint stabilization and movement. And another thing to realize about tendons is they're relatively avascular. So that's going to what that's going to tell us is, as far as from a hormone perspective and everything, how the tendon's actually going to go through its natural remodeling phase. So the tenocyte is responsible for producing the components of the extracellular matrix of the tendon. So that's the fibroblast responsible for actually making up the components of the extracellular matrix. The tendon is going to be reliant upon the autocrine and paracrine response in order for the enzymes and hormones responsible for remodeling the tendon are going to be taking place. And that's going to be primarily due to the fact that you have a relatively avascular structure. So remember what we talked about at the beginning of the discussion here. We had mentioned how, the amount of blood flow that comes to a structure will dictate how much the endocrine system actually will directly affect it, because obviously the endocrine system is going to carry the hormones to the area via the blood flow. Well, in the case of the tendon, you're not going to have that. So it's going to be a little bit more reliant upon the autocrine and paracrine process in order for that to take place. Now, as far as the factors related to pathology in the tendon, here we're going to be talking about disruption of the normal mechanobiologic response. So when we start to talk about inappropriate loads to the tendon, so in other words, we're either overloading the tendon by doing too much of something or having some type of biomechanical problem which puts added stress on the tendon, you get this inappropriate load. Now, one of the things that does tend to happen with that is you do get alternate phenotypes of the cells. So we said that the tenocyte's responsible for producing most of the components of the extracellular matrix in the tendon. In the case of a pathologic situation, you actually get chondrocytes, adipocytes, and osteocytes. So those are the cells that actually make up cartilage, adipose tissue, and bone respectively. We actually start to see that happen when tissues are put under an inappropriate load. So mechanical load leads to the production of neurochemicals. So, one of the neurochemicals they believe is primarily responsible for a lot of the pain we experience with a tendon condition is what's called substance P. So substance P is a pain chemical that gets released. In addition to that, during a disruption of the normal anatomic process of the tendon, you also get the formation of the substance P sensitive nerves that start to form. So they're nerve fibers that will actually form in response to this. So while two things to look at when you look at the pathology of tendons. So one, you can just get a strict disruption of this, the normal mechanobiologic process that tenocytes use to create extracellular matrix, or you have another situation where more of it involves tenocyte-produced neuropeptides. So that would be those chemical mediators that occur that produce that pain and discomfort. So we think that one of those two, or maybe to a degree, either both of them are the issues related to, when we see clinically a tendinopathy in an individual, or we see that degenerative process take place. Another important thing to note is that the tendon cells communicate throughout the tissue via what we call cell junction zones. Okay, so this intercellular connectivity, these cells communicate through each other as far as signals to kind of regulate the normal homeostatic structure of the tendons. That's the way that the cells communicate. Okay, so, and the cells communicate. The protein responsible for that is what's called a connexin. So that's a protein that allows for the communication between the cells within the tendon under normal conditions. And that's another thing that can get disrupted by, again, some type of abnormal loading. Okay, so, and again, when we're talking pathology to the tendon, again, kind of the broad general term is what's called a tendinopathy. Again, the term tendinitis gets used a lot, and you can actually develop an acute inflammation of the tendon. However, what is typically seen clinically is more of a breakdown condition. Okay, so again, some type of abnormal loading occurs to the tendon and causes what's called a tendinosis, where the tendon actually it's a degenerative process that happens where the actual structure of the tendon starts to break down and the makeup of it actually changes. So this illustration's in your textbook. You can see here, so on this side, we have what a normal tendon would look like under a microscope. Over here, this is a pathologic tendon. So if you just notice the difference, what this is essentially showing is this is showing kind of like the natural collagen fibers and their alignment in between. You could kind of see within here the ground substance, whereas over here, you just kind of see this more or less a mess of fibers. They're not aligned. It's kind of all kind of randomly placed throughout. So again, there's kind of still debate on the actual like factors as far as like how tendinopathy actually happens. Now we have some pretty good ideas just based on physiology and some of the things I already talked about. So here we have what we typically think of as the classic mechanobiologic response due to an abnormal load. So this is one of the theories that we think happens, where basically just the overall tendon structure gets altered and disrupted. So what happens is due to some type of, again, abnormal physiologic stress, be it through someone's biomechanics or just inappropriate loads, you start to get changes in the extracellular matrix, which changes the mechanical signals as far as constantly being able to do the normal remodeling of the tendon. So what ends up happening is you basically encourage the development of this degenerative tissue in the tendon. So the mechanical overload starts basically a signaling cascade of enzymes that produces this abnormal thing. So this is kind of what we talked about a little bit with the different cells that form. So your textbook, and the percentages aren't that big of an issue, but your textbook talks about how 4% strain basically causes tenocytes to basically produce and represent the extracellular matrix in the tendon. When you increase those loads, you start to get the markers for those other cells that start to form. So again, I talked about the chondrocytes, the adipocytes, and the osteocytes. They will start to come in the area, and the actual formation of the tissue will start to change. So that's one of the areas that we think would contribute to, or one of the ways we feel that there's this abnormal remodeling of tendon due to these either an abnormal load, meaning that the tendon just gets loaded too much without getting the chance to remodel, and or some type of, if you look at biomechanical problems that people might have, postural issues, that would put the tendon under undue stress and cause this to happen.